My name is Coralie Viollet. I'm leading the genomics lab at Boehringer Ingelheim in Germany. Together with Santacruz, we support the RESOLUTE consortium by assessing the transcriptomes of different cell populations. Our aim at RESOLUTE is to systematically de-orphanize solute carriers by utilizing and combining different omics approaches. Using transcriptomics approach, we want to better understand how gene expression is altered when a particular SLC of interest is either expressed or not. We can then start to map out the downstream effect of that particular solute carrier onto key genes and pathways.
We've been able to establish gene expression patterns for known SLCs with known substrates and known functions. And then when looking into an orphan SLC, we can map the transcriptomic changes to our established gene signatures, which may indicate that we're dealing with a zinc transporter, for example. Transcriptomics is only one of many omics methodologies used within RESOLUTE. When integrated together with ionomics, metabolomics or proteomics, we really start to build a more comprehensive picture of what these proteins are doing individually, but also as a family. Our lab has generated more than 5,000 bulk RNA-Seq samples in the past 3 years, and we will make these results accessible in the RESOLUTE website, while the raw data will be deposited into public repositories. Now Coralie will give you more details about our scientific approach.
At RESOLUTE, we use genetically engineered human cell lines that have an SLC either deleted or overexpressed. If the transporter is no longer transporting its substrates or there is too much of it, the cell will trigger some compensatory mechanisms to circumvent this. After we extract RNA from these cells, we enrich for messenger RNA. We then fragment it and do a reverse transcription into single-stranded and then double-stranded cDNA. We then ligate some universal sequencing adapters at each end, and we PCR amplify the product together with unique primers for each individual samples. We will later be able to pool these samples together for sequencing on the NovaSeq instrument. After sample processing and data analysis, we compare genetically modified cells versus wild-type controls to reveal which genes are changing upon deletion or overexpression of an individual SLC. With this transcriptomics dataset, RESOLUTE wants to empower scientists worldwide to further investigate solute carrier transporters.